from this, I would like to invite our next speaker, who is Gilles Atlan. He's Vice President of NF2 Biosolutions, and he's very well known to NF2 community. Gilles, thank you so much for joining us, and that's definitely an honor to have you here with us today. You're a prominent promoter of various innovation therapies, and also for those who don't know, Gilles has a daughter who has NF2, and so he's a very active dad who's like acting to promote NF2 and to bring knowledge. And he's going to talk about latest developments in NF2 research. Gilles, you're very, very much welcome. Thank you for joining us. And the floor is yours. We can see your presentation and hopefully we can hear you. But if not, I will talk. Thank you. Yeah, we can hear you.
Thank you very much. Thank you. So thank you for the introduction, and I'm happy to give you an update on the ongoing research. I'm not going to talk about all the research that are happening currently, more the one that we are involved in, but not only. So I hope that this will give you a lot of hope. The only thing is like research takes time. The process of reaching a drug is quite a very slow process, but at least we have a lot in the pipeline. So I'm the father of Karen. Karen, she's today 17 years old. She was diagnosed when she was 6 years old with NF2. By the way, we thought she had NF1 at first when she was newborn, but she was misdiagnosed, even in a very good center, a genetic center that was at Stanford, California. We were living there at that time, but as it was told before, there is a lot of lack of knowledge and misdiagnosis regarding NF1, NF2, NF3. So at the end, at 6 years old, we were sure that she has NF2. The MRI confirmed it. And a pretty typical NF2 case for her. She has bilateral vestibular schwannoma on both sides. As you know, the vestibular schwannoma is called also acoustic neuroma. And the problem with them is like it disturb the balance, but also you can lose hearing over time. And also it can, if you continue to grow, it can hurt the facial nerve, leading to a facial paralysis. And the facial paralysis can lead to paralysis of the eyelid that can lead also to lose the vision, even that there is also cases of tumor on the vision nerve, on the optical nerve. But classically, for most of the NF2 patients, they have bilateral vestibular schwannoma, and then they have schwannoma, peripheral schwannoma elsewhere in the body, often along the spine. In the case of Karen, she has many along the spine and other little, also meningioma is for almost 50% of the patients, they have meningiomas. Here you can see on the photo, you can see an example of a vestibular schwannoma here, and a peripheral schwannoma right here next to the spine. So currently what we tried, now she's 17, but when she was 11 and then 14, we did 2 surgeries to try to remove the vestibular schwannoma. The problem with this surgery, it's very dangerous. It can, I mean, it can, you can lose the hearing because of the surgery. But we also know that if we don't do anything, that usually the tumor continues to grow and then you are going to lose the hearing. So in her case, she was lucky, she could keep the hearing, but we never know if the tumor will regrow or not. And that happens often, that after surgery, after a few years after surgery, it can regrow. So there is no real answer for NF2. Even if a surgery is well done, it can always regrow at the same area. If you do radiation, it impacts the nerve as well over time. And as you know, there is different inhibitor drugs that are on trial, but these are repurposed drugs that are used for other cancer, and usually you cannot take it for so many years. And since it is a genetic disease, it's not a virus that you get rid of it. You have to take, if you use an inhibitor drug, you have to take it over a long period of time. So that's why we created NF2 Biosolutions, that is a patient-led non-profit, in order to start innovative approach on NF2. We saw that there is a lot of drug repurposing inhibitor going on, and that's good. It could be an answer, or it could give us time until we get a long-lasting drug for NF2. That's why we started different kind of innovative drug. What we did, we look for the researcher that are having an innovative platform for treating a genetic disease, genetic disorder, so the disorder that appear because of a specific gene, or specific protein that is not produced. And then we try to apply that for NF2, and that's why it led us to focus more on gene therapy and immunotherapy. And there is different kind of gene therapy and different kind of immunotherapy as well. So many different approach. So far, we have raised $2 million in small donations over time, and this money was invested in this different research, preclinical research. I will talk about it later, but as a foundation, we are able usually to fund preclinical research where they do their test on cells, on cell line, or on animals, on mice. But afterward, to do the test on human being, that's require way more money, and that's where usually private funds are raised in biotechs for it. So we could fund 11 different labs on different approach and tools, and I'm going to go through most of them during this presentation. So the challenge with NF2 is that the patient has different needs basically, and it depends more how the disease is progressing for them, and then at what period of time they are. If they are really at the beginning, if they are very young, or if the tumor are very small, maybe then their need is just to stop the growth of the tumor. And then the tumor didn't do any damage yet, and they could, if they stop the tumor growth, they will be happy with it. For some, the tumor is already dangerous. It's already pressing on the brain, for example, or on the spine. So they need to destroy it to remove the tumor. And sometimes the surgery is too complex to do it. So there is the need also to destroy existing tumor. So there is the need of destroying existing tumor. For some patients, they have the need of stopping existing tumor growth. And there is the one that don't want any new tumor creation to happen as well. Usually, if you know that a baby, a very young person, has NF2, you don't want, and usually the tumor are developing over time in the first few years. You start to see them appearing. You don't want any new one being created. Even after the people that have a treatment or have a surgery, you don't want a new one to appear over time. And then we sponsor different research, and here we assume that these different, are not sure yet because we have to see really the effect on patients, but we assume that some of these drugs are more focusing on reducing existing tumor, on stopping growth, or avoiding new creation. So let's start with the, and then we will go through each of these approach. And then we have also 3 different tools that these labs need to use, and we sponsored the development of these tools as well. So the first one is bacterial immunotherapy. That's the most advanced research that we sponsored. That has been started at Harvard by Dr. Brenner and Michaelanos at Harvard Medical School. And they used a bacteria, genetically modified bacteria, to create an immune response inside the tumor. An immune response, it means it will attract your immune cells to that tumor, and then the immune cells will detect that there is some tumor cells, and they will attack the tumor cells. So on mice, you could see a reduction of size, and also they stopped the growth and also started to reduce the size. But since it is immunotherapy in that case, because we use a bacteria to trigger the immune response, in mice, they saw that if they inject inside one tumor, the other tumor also are decreasing, because then your immune cells in your body are starting to understand what is a tumor cell, and they can find others in the body. So now it's currently in the hand of a biotech Mulberry that are working hard at raising the funds to run a first trial, and we are helping them out as well in order for them to find the necessary funds. So that's the the most advanced one, because once they have the fund, they can start right away a trial. Then we have also another technique called the Susan gene therapy. So it's not immunotherapy. It's not that we are not triggering here the immune response. We inject a gene that creates the cell death of the tumor. So here in theory, it will reduce also the size of the tumor. And that's also done by Dr. Brenner at Harvard Medical School. So we are pushing this research forward as well. So again, it's using a AAV delivery mechanism, that's a virus, to deliver a gene that creates the death cells of the tumor cells. It's called apoptosis. So a lot of technical term, and I'm not going to go through this technical term, but I just want you to understand the idea behind it. Here we want to destroy existing tumor cells. Then another one that we founded from the beginning as well, that we jump started from the beginning as well, is gene addition therapy. So it's also using a virus to get to the cell, but here we want to restore the creation of myelin protein. So why people have NF2? NF2, they have NF2 disease because the NF2 gene is mutated and doesn't produce the myelin protein. And when it doesn't produce the myelin protein, the tumor cells don't know that they have to the skin, the cells around the nerve, they don't know that they have to stop to grow, and so they grow. They continue to create cells, Schwann cells, and then it creates a tumor. They don't know that they have to stop to create Schwann cells because they don't have this myelin protein. So here the idea is to use a virus to inject a functioning NF2 gene that will generate this protein, this myelin protein, and in order to stop this multiplication of Schwann cells that create the tumor. So that's at the lab of at Nationwide that was started with the Dr. Mayer labs, and now we received a grant from the NIH to do the toxicology study, and we also created a myelin therapeutic biotech in order for Dr. Noah Young to raise the funds necessary to move forward this approach. But we have the proof on preclinical study on animals that this can work. This could work. We can restore myelin creation in the NF2 in the cells, and then it stops the growth of the tumor. And then maybe over time, the tumor cells will also die off, so it could also reduce the size of the tumor. Then we have also non-viral gene addition therapy. So it's all here also we add the NF2 working NF2 gene to create the protein, but we don't use a virus. In that case, we use something called antibody. And the advantage of this antibody is that we don't have here for sure any problem of redosing over time, because with the viral with the AAV is used a lot in other drugs. But there might be an issue of redosing many times, but with antibody, that should not be an issue. And we have been testing that on mice, and we could see that this is going to work. And one advantage of this antibody approach is that it creates also an immune response. So a bit like an immunotherapy response. So it's not only delivering the gene that produces the myelin protein, but also it generates an immune response that attracts the immune cells to also attack the tumor. That has been proven also in other research. By using only the antibody, they could see an immune response. And also it could potentially we can also use that delivery mechanism to deliver also an inhibitor at the same time. So we could do three aspects. So the immunotherapy, delivering the NF2 gene for myelin creation, protein creation, and also an inhibitor. And so that's in the hand of Dr. Zhu at Yale University. And we are currently fundraising for this project. He's been working on it already for 2 years. We sponsored it from the beginning. And now he's creating humanized antibodies that he's testing different kind of it to see which one is the most effective, currently still on mice experimentation. We did also a pilot, a gene editing pilot. So that's editing the gene. Before it was adding a gene, but simply adding a functioning gene. Here we are editing the existing gene. We just wanted to see if, because that's what's going on also with other type of disease. We wanted to see if that was possible with NF2. But the challenge with the NF2 is that people have many different type of mutation in that NF2 gene. So it's more like a specialized drug for specific type of mutation. But Dr. Chernoff at Chase Cancer Research could prove that this is possible to take care of some type of mutation, like large deletion, we are also fundraising for research team in Manchester University. They have a very good NF2 clinic as well over there, and we are through our NF2 Biosolutions UK, we are funding the preclinical research over there. They have a great team, and we had Grace, Gregory, and Adam Jones working on it since 2021. Some of the different type of research. Now there is a new group of research being done for NF2, but these are results from the first one that they started. They identified that between a sporadic vestibular schwannoma, it means people that have a vestibular schwannoma, but they don't have NF2. They simply have a vestibular schwannoma appearing, but they don't have an NF2 mutation in their whole body. So and that's way more common. It's happened usually later in life. There is only one. It's only on one side, not on both sides like in NF2. But there is way more patients with this, and they could prove that they are very similar. They analyze all the different cells that are in the vestibular schwannoma, and they see that they are both very similar. The sporadic vestibular schwannoma is very similar to the NF2 vestibular schwannoma. And this is important to prove, because when you want to raise funds for NF2, you can say that if we get to a cure, this cure that's work on these specific cells could work also on sporadic vestibular schwannoma as well, depending on the type of approach, because they are very similar. They have the same type of cells. So the microenvironment of both of these type of tumor are the same. That was one of their discovery. And they are working on many other aspect of it as well, about the T cell, that it is related to immunotherapy, on what should be done, which T cells need to be increased or decreased in order to stop the tumor growth. And the macrophages, that is another type of cell that you have in the microenvironment of the tumor, how to regulate them, what's the impact of the macrophages on the tumor growth, and how they could how they could manage the macrophages in order to reduce the tumor growth as well. So we have a big team of people over there that we are sponsoring, research students and PhDs, and they are working already for a few years. So what is great there, because we really ignite the research on NF2 over there, and now it starts to roll, and now they are looking for funds from other sources, not only from us, from other sources, and they increase their team, and they are working more and more on NF2. There is also, we are not involved directly in inhibitor drugs, but I wanted for the patients here to let them know that there is also research being done on inhibitor drugs. The one that finished not long ago is the trial of brigatinib done by Dr. Plotkin in Mass General. And now many people, from what we know, and my daughter as well, are using brigatinib, hoping that it can slow down the growth of the tumor and sometimes decrease it. So that's another option that you have today. I know a lot of people are using Avastin, but sometimes Avastin is more recommended when you already have hearing loss and you want to try to restore hearing loss, or you already have no other opportunity, no other way to deal with the tumor with radiation or surgery. But usually it's good when you have a hearing loss and you can restore it sometime in some cases for you. But now there is also brigatinib that if you didn't reach maybe the hearing loss, or if maybe Avastin didn't work, you can try it as well. And that's inhibit. So there is many signals, many pathways in tumor development, and one of the pathway is called ALK. So this brigatinib inhibits the ALK pathway. These are some screenshots from the paper that has been published about the research, and you can see that on the right side, that's the numbers are how much in percentage the tumor decreased. So 10%, 20. So above 20% is really more like a categorics, because since MRI measurement is not accurate, they really want to consider only the one that are above 20%. So you can see quite a lot of tumors. These are tumors at patients that decrease, or at least they didn't grow. So that could be something for you as well. A lot of people start to take it in US as well. Then we have also translational research tools that we developed. So I will go very quick on that one. So we had NF2 Biobank, because this researcher, they want to have access to tissue, to cells, to cell lines. So we started to collect tissues and cell line, or make cell lines from patients. So we can provide this to researchers. We have also, we didn't have a mice model for epidemoma. Epidemoma is the third type of tumor that NF2 patients are developing. NF2 patients develop schwannoma that are vestibular or peripheral schwannoma, but meningioma as well, and epidemoma. That's a bit more rare, but there was no mice model. So when you develop a drug, you cannot test it. You cannot see if it works on an animal, because there is no animal with the NF2 type of epidemoma. So we founded a research at Salpêtrière Hospital in Paris by Professor Michel Calmarides, that, by the way, is running an NF2 center, NF2 patient center. He's a clinician, he's a surgeon, but he runs also the NF2 center, and he's doing a lot of research on NF2. And then also a good, very good project that we supported is the ability to automate the volumetric, the measurement of the vestibular schwannoma of the patients. As you know, when you do an MRI, the measurement is not accurate at all. In your MRI report, you just get X and Y dimension, and you think it doesn't grow, or you think it grew, or it decreased, but it's not accurate. You are never really sure that it really grew, because maybe the slide, the photo where they measure the size, maybe it's not exactly at the same place that it was compared to the previous MRI. So it's really not accurate, and you have to know the growth rate in order to be able to do a surgery, do a radiation, or take a drugs, or really telling you that this disease is not stable, it's growing. Sometimes you think it's stable and it's not, or the opposite. So for example, this is an example. On the left side, you have the typical linear measurement of a tumor, what the type of measurement you get in your MRI. So here for that person, it says in 2016, it was 1.5 centimeter times 2.3. Then 1 year later, it shows that it decreased. So the patient for sure was very happy about that. But when you do the volumetric study, when you do the volumetric calculation, it means the computer with AI, with artificial intelligence model, it can identify the vestibular schwannoma and calculate the size of every slice, every photos of the MRI, and then calculate the whole volume. It showed that it grew by 33%. It didn't even decrease, it grew by 33%. And that can influence a lot your decision. Here, for example, another example from 2021 between these two MRI, it showed that it's quite stable, but no, it grew by 20%. So it's very important for us patients to have access to this volumetric measurement. I personally have to pay a labs to do it for my daughter, so I can know exactly. This is a screenshot of the tumor of my daughter's. so I can know exactly the growth rate. Here the red went down because she did the surgery. Here it was growing, then we tried everolimus drug. It's an inhibitor, so it worked a little bit at the beginning and then it grew again. But at least I know that the size is well measured, because this is what helps you to decide about what to do next. For example, Dr. Slattery from INE, from the House Clinic in Los Angeles, he decide what to do based on the size of the tumor, but you have to know the real size. You have to know if there is no growth, or if there is a growth no more than 2 millimeters, because that's how he decide what to do next. So with this AI model that automatically calculate the growth of the tumor, but also you can see which area of the tumor grew. For example, the yellow area is the part of the tumor that grew. So the main challenge for us and for research is to raise the funds for doing the first trial. Once there is a first trial and there is some positive data, then it's easier to raise funds. But to have this first trial on human is costly, and a lot of people don't want to take the risk of putting money at that stage. As a foundation nonprofit, by raising donations, we are able to raise money for doing the pilots and preclinical research. But for raising money for trials, that demand a few millions of dollars, and this is challenging. So if you think about any in your network, anything that could help that phase, maybe someone that is very wealthy, someone that could be interested in this different research and sponsor them, let me know. So far, through our nonprofit, we raised money mainly from NF2 parents and NF2 family, and they are using their network to raise these funds. Money can come from them, but also from their network and where they are very active. So we are really, really dependent on the NF2 community, the parents and family members. We are making a lot of webinars on different topics that are more clinic. So how to manage your own disease, what's happening with the vision, with the surgeries, with the Avastin. So we have very good webinar from where the expert in the domain are talking in this webinar, and we have also webinar updates on some research as well. So you can find them on YouTube at I Need a Cure, and also on our website, NF2Biosolutions.org. You have a tab there of webinars, and you can find all the webinars. Very, very important. And now you can find any languages. You can change the language on YouTube, and you will have the translation. And it will be a very good translation, because we took the old text, and we did ourselves the translation. So all the scientific words are kept and are accurate. Thank you so much, and I will be happy to answer to some of your questions. Here you have my email as well, if you have questions you would like to ask later offline.
Gilles, thank you so much for your presentation. I suggest in this room, at least, we give him a warm round of applause.